Now, in addition to the counterbalance of the deforming force, there are even few other purposes for embedding. What is that? Normally, when we collect a tissue, in the tissue, we find multiple cells, right? We already know that tissue is made up of cells. That means if this particular tissue be collected, there will be cells inside this tissue. And the cells present inside this tissue naturally would overlap over each other because we are collecting a mass. Right. Now, in order to prevent this overlapping of cells in a tissue, or the extracellular matrix, that means if these are the cells, then surrounding the cells, we can have spaces that I'm showing in yellow color. This can be considered as the extracellular matrix. So overlapping of extracellular matrix or overlapping of the cells of the tissues can be distinguished if the tissue is embedded in a proper medium. Because in embedding, what will happen, part of the paraffin would enter into the spaces in between the cells, that means in the extracellular medium. Clear? The alcoholic treatment that we did for the dehydration basically acted upon these cells, right? Because the water content that we are speaking of is distributed within the cells mostly. But the extracellular matrix is having a relatively less amount of water. The result is less amount of replacement of water will occur in the extracellular matrix. So the xylene treatment would also be less effective in the extracellular matrix area, and thus paraffin can get infiltrated into this extracellular matrix. Understood? So what we need is the infiltration of the paraffin into the ECM. Now, individual cells would have the coating of xylene. Having the coating of xylene means paraffin cannot enter into the individual cells. Clear? So I here have explained another utility of embedding. So for embedding, the details that we need to take is first the common embedding material. What is this common embedding material? This is paraffin. Now the paraffin as the embedding material is used when the microscopy would be the light microscopy. That means for electron microscopy or say transmission electron microscopy, the paraffin is not used as the embedding material. Understood? Now, the most common microscopy is the light microscopy because it is relatively the most cheaply available microscopes. Now, being light microscopy the most common, so paraffin as the embedding material is thus also the most common. Right? So how do we embed this? We embed the cleared tissue, so the cleared tissue is placed in molten. This is another advantage of paraffin. The pins, by very little amount of heat, you can make the paraffin to melt. It's a kind of waxy material, right? Mombati moto, right? So little amount of heat can cause it to melt. Now you cannot subject your tissue, your sample, to huge or heavy heat, because if you're subjecting your sample to a heavy heat, that heavy heat itself have a deteriorating effect on the tissue itself. Hot will destroy a tissue, but naturally it can even burn the tissue. So you need that particular material as embedding material, which can melt easily with slight amount of heat. And paraffin can be melted with slight amount of heat. Now, after you apply slight amount of heat to this paraffin, it will become molten. So once it is melted, you can place the tissue, you can place this clear tissue into the molten paraffin and leave it aside so that this molten paraffin next solidifies. As it solidifies, it retains this cleared tissue within it. Are you understanding? Normally, what we use are some blocks. If say these are two plane surfaces, accordingly we can have another plane surfaces covering from all the sides. So this is how a hollow block is produced, right? You put this tissue into this hollow box. After putting the tissue into the hollow box, put paraffin there. Which paraffin? The molten paraffin. So if you're putting the molten paraffin in the hollow box, leave it aside. So the molten paraffin now solidifies. Now once solidifies, these plates can now be removed. And after the removal of this plate, what do you get? This block that entirely is made up of now solid paraffin. How it's now a solid paraffin? Because by leaving the paraffin aside, the molten state becomes a solid one. But the thing is, now in this paraffin block inside, we are having this cleared tissue that we are labeling as T. Understanding? Now we can apply our knife over this block so that sectioning becomes possible. Okay. So what was the process that I discussed? Let me repeat. We are first melting the paraffin, and into that molten paraffin, we are placing our block of tissue. Right? After placing the block of tissue, we are covering that block again with paraffin. So when we are initially preparing this block with the help of these plates from all the sides, first we pour some amount of paraffin. So let me numerically order these steps. Pour paraffin, molten, not to the full, but only in part. Because if you completely fill up this block with molten paraffin, the tissue that you want to embed would remain floating over the surface of the paraffin, right? Liquid paraffin বা মোমেতে কি হয়, মোমের উপরে একটা কিছু যদি দিয়ে দাও ওয়াক্সের উপরে, would that material dissolve into the mass of the molten material? No. Right? So this block preparation would be in 3 steps. The third step is again to pour the paraffin, that is molten paraffin, till the brim of the temporary block made with the plates. Understood? The plates that we were using to create the hollow space, part of the hollow space to be filled up with the molten paraffin. Remaining part would be again filled up with the molten paraffin, but in between this, there now is the second step. In this step, what are we doing? We are placing the tissue. That means what? If I draw this block again, to some height, you first place your paraffin till some height. That means the first step is over. Thereafter, in the second step, you place tissue. Then, as the third step, you now cover the remaining volume again with molten paraffin. That is how this entire volume is now having the molten paraffin. Set it aside so that the paraffin now solidifies. And once solidified, remove these individual plates. And as these individual plates are removed, what we are now left with is a solid block of paraffin, inside which there is the tissue. Understood the concept?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Now, in order to retain the sharpness of the edge of a paraffin, I mean of the knife with which we are going to section, the next step is what? The next step, the fifth step, would thus be here written as sectioning. So what you do is, after you create this block, you can trim some parts of this block. Some very superficial parts of this block can now be trimmed. Why do we need to trim it out? So as to reduce the thickness of the paraffin material outside the tissue. The more the thickness is removed, the better the sharpness of the sectioning knife is retained. Are you understanding? So after removal of some edges of the solidified paraffin, we are now left with a rough shaped paraffin. With this blue, I am representing the paraffin, inside which there is still this tissue present. Here. Now we need to mount it up. That means, even before sectioning, we would now need to mount. Sometimes mounting can even be done after the sectioning. The purpose of sectioning, I mean mounting, is what is easy handling of the tissue. Clear? That is the purpose of mounting. And the function of sectioning is to bring out thin slices of tissue to be removed from this block. Okay, the machine that we use for sectioning is known as microtome. There are many different types of microtome. There are many different blades used in the microtome. All those details are to be discussed in the upcoming semesters when we will discuss more detail about the histopathological studies. As of now, just know that the microtome is the tool that is used for sectioning the tissue. Now, the section that we require for optical microscopy or the light microscopy should be around, say, 5 to 10 micrometer thick. If the thickness increases, then there is a chance that you would not be able to see the tissue under the microscope. I already explained in one class that more a tissue becomes thick, the less the tissue would allow light to pass through. And as lesser amount of light passes through, so it becomes even more difficult for you to observe the details of the tissue, the structural details of the tissue. Right. Now, this thickness is sufficient for a light microscope. If the slices be even thinner than 5 micron, then the chances again would be that gross details would get lost. Gross details. See, with light microscopy, we start with the lower magnifications first to observe the gross details. Only after the observation of the gross details are over, then we move on gradually to the higher magnifications to get the finer details now. Understood? That is the process. We start, say, with 10x. Thereafter, we can move on to the 20x for a bit more finer detail. Then we can move to the 50x and thereafter finally to the 100x. We can even move on to even higher magnifications. Now, more higher the magnifications, that means the broader or the larger details are getting lost. The more we move to the higher magnification, the more thin the slices are required to be made. If we have to stick to the gross, then this amount of thickness is sufficient. Even at very high magnification, we do need to even change our microscopy technique from the optical microscopy to the electron microscopy, in which the sections are even thinner than what I have written on your screen. Are you getting the point?
Yes, sir.
Now see, what is required for sectioning? is that the material to be sectioned becomes hard. Now, for hardening, we can have solidified paraffin, as we have already noted. But apart from that, sectioning can also be performed on specimens that are frozen in liquid nitrogen, or there is a technique of rapid freezing. Rapid freezing technique is there. Rapid freezing technique, if be followed, then we use another method of sectioning, and that method is known as, the tool is known as cryostat. Okay, so in the cryostat, what do? We use a pre-cooled steel blade. Why do we use a pre-cooled steel blade? Because the temperature in that case is never allowed to rise in between the steps of embedding and the step of sectioning. Understood why that steel blade needs to be pre-cooled. And these sections, for the purpose of microscopy, when they are mounted or when they are placed on pre-cooled, again, glass slides. So these are the techniques that we are performing when the method of embedding is not the usual one, but the method of embedding is this frozen liquid nitrogen or a rapid freezing method. See, freezing is one technique of preservation of structure. Exactly for that reason, in our home, we use refrigerator. So refrigerator can preserve the structure, say for example, for meat or for fish for some duration of time. Though at that same time, it can cause some amount of drying. Actually, that is not a drying, because for drying, water needs to evaporate. But the drying, we sense it because, inside the refrigerator, there is also dehumidification. The humidity is less. So some amount of water is evaporated because the humidity is less, and the remaining amount of water, instead of being evaporated, becomes fixed by freezing. So if anything, if a fluid is frozen, that means it then becomes fixed. So that is another method of fixation. There is even heat, microwave, all these methods of fixations are available, which will be detailed again, as I was telling, on the next session. As of now, since not much time is left for this class to get over, let me give you the idea of the last step, staining. On the next class, I will discuss few individual stains, and along with that, I will mention the color that each part of the cell takes once being treated with this individual stains. Usually for the light microscopy, we use water-soluble stains. Light microscopy, we are using water-soluble stains. So most of the stains that you find in the laboratory are water-soluble. And when we buy the stains from the market, we buy it in the raw form, because it is not at all a point to spend for water. Water is a relatively more common available source or available resource, right, in the laboratory. You do not need to buy water in huge amounts. I mean, when you are spending for a stain, do consider spending to be utilized by purchasing only the raw part of the stain. You can prepare your stain by dissolving the raw stain in water, because for light microscopy, we use the water soluble stains. Right. Now, one thing is necessarily to be performed before we ensure that the tissues are getting sufficiently stained. And that thing is the removal of paraffin again. See, the paraffin was necessary for sectioning. But once we have already sectioned the tissue, if we still retain the paraffin, then that retained paraffin would interfere with this water soluble stain, because we know that paraffin itself is not water soluble. Are you understanding? So putting the diagram again, if this be the tissue, surrounding the tissue in the green color, I was showing this paraffin. If this paraffin is retained, if say there are multiple stains on the outside, some of this color, some of the blue color, none of the stains being water soluble would be able to come in contact with the tissue because of the paraffin. So the paraffin needs to be removed from the mounted sections. Okay. Now, after the removal of the paraffin, what do you need to do? In cases, sometimes we do again need to rehydrate. You might be confused that if you are rehydrating, why in the previous step we dehydrated the tissue? The previous step of dehydration had another purpose. That is, if we are not dehydrating the tissues, the successive stages of embedding and thereafter sectioning cannot be performed, because for sectioning, we need to have a hard substance. If water is retained in the tissue necessarily would remain soft. So in order to ensure that the tissue becomes partly hard, we do need to dehydrate. Now, after dehydration, we have hardened the tissue. After that, we have sectioned it. After sectioning, when we now need this water-soluble stain to enter into the tissues, occasionally, this step of rehydration becomes a necessity. Once it is to an extent rehydrated, it's now easier for the stain to enter into the tissue. Did you understand?
Yes, sir.
Yes.
Yes, sir.
Now, after staining,
Yes, sir.
we have stained the tissue. After staining, this water that was introduced in the rehydration steps can again be removed. This has to be done as a necessity. If rehydration is a step, then dehydration would again be a step. Because if we are not dehydrating that tissue, we would not be able to retain that tissue for longer duration of time. See, we have already sectioned it. So sectioning means what? The tissue has become now very fine. The surface area of the tissue has increased. So as the surface area of the tissue has increased, whatever the rehydration that we have made on the tissue would now get evaporated naturally. In order to prevent that natural evaporation, we don't need to dehydrate it artificially. After we dehydrate it artificially, then we can put something that is called cover slip. I think you have noticed sometimes it comes in square shape, sometimes it comes in a circular shape, a kind of glass piece. very thin though. Once we put that cover slip, that means all chances of further evaporation is now made to 0. So the cover slip protects the tissue from being damaged, as well as the cover slip becomes necessary when we view the tissue under the microscope. Why? Because consider that this is the slide and we have put this tissue onto the slide. If we are not using these cover slips, in that case, the objective of the microscope can accidentally come in direct contact with this tissue. It can happen, right? So if it ever happens that the objective is coming in direct contact with the tissue, then that direct contact can damage both the tissue as well as the objective lens. So in order to prevent the damage, it is always better to cover up the tissue with a cover slip. Understood? Why is it necessary?
Yes, sir.
Okay. So now your tissue is prepared. You can now put this slide under the microscope and can carry on with your observations. Okay. I will have to describe few stains now. The basic classifications of the stain, and for that I will be taking another class that will be on the next week, on which some common stains and their chemical affinity would be described and discussed. Okay.
Yes, sir.